Okay. Yeah, so I'm on like, I think 13 or 14, number 13 or 14. So I have like, I think I started grading 13. So I have 13, 14, and 15, and then I'm done with it. All right, what am I doing? I need to join on Zoom. I got distracted.
Sorry.
No, that's okay. It's okay. No, I'm not rating this now. Okay, share content, screen, start broadcast. Come on. Okay, put do not disturb on. Yeah, that would be good, right? Okay, I think we're ready. And I have iCloud up, so check in on iCloud, please. That's why I'm putting it up there. iClicker, iCloud. Well, so for me, it's called iCloud. For y'all, it's called iClicker, which makes it really.
Oh, because you have all of them. Huh?
Because, well, yeah, because and all my, yeah. That's like the professor side of it. So, okay. And I hate seeing myself. Please hide that. Please hide that. And I'm like listening to myself or seeing myself, right? All right. So why should you never play hide and seek with a leopard? Because you'll always be spotted. You'll always be spotted. And then the second joke that I gave my son today, he wouldn't even tell me. I just put the cards in his lunchbox. He wouldn't even tell me. He was like, Mom, I already did that joke. And he wouldn't even tell me what it was in the car. So I'll only get one today. He's gotten real picky on jokes. All right, so we're gonna finish 17 today. We were like almost finished last time. We're gonna start 18, but I'm not going really far into 18 because I'm like super exhausted. I'm gonna be honest. So that's my plan for today. And then I'm almost finished grading your exams. So hopefully by Thursday, I'll have all your exams back to you, all your case studies, so that we are 100% up to date before drop day. Yes. That's my goal for all of my classes. Oh my God. Lab is the hardest. Lab is the hardest, right? Yeah. All those lab reports. All right. So we talked about heme biosynthesis, right? Then we talked about breakdown. So when we have all these steps in heme biosynthesis, what did we say was the common disorder that, like the common name for the class of disorders that results from dysfunctions in heme biosynthesis? Remember? Not hemophilia, close. Porphyrins, the porphyrins. So porphyria.
Really?
Yeah, porphyria is one of them. So remember, well, you don't remember, but maybe in your history lessons, you remember reading about the Mad King, the King George, who was the king during the Revolution, American Revolution. Is it the third? I don't, maybe. You might have to look it up, but like 1770, somewhere in there into the 1800s. So he was called the Mad King, right? Why was he called the Mad King? Because he would have these like periodic episodes where he was like crazy, right? But he also had really dark urine. So one of the commonalities in all of the disorders in heme synthesis is that you're going to have a buildup of some discoloration in the urine because of heme, right? Heme carries what? Well, heme carries something that binds to oxygen, iron, right? Exactly, which is dark. So a lot of times you would have dark urine. That's a good indicator that something either in heme biosynthesis or breakdown is wrong. And so it's really interesting disorder because it can be sporadic and it can be triggered by environmental cues. So whenever he would start to have an episode, they would give him arsenic Awesome
Gotta point me
treatment,
down
right?
one way.
Probably not the right thing to do because it actually exacerbated his condition, made him even crazier, right? So maybe the American Revolution would have gone differently if they had treated him with something different, but you know. Anyway, okay, so then we talked about bilirubin, right? So when we talk about heme, right, now that we've made heme, because heme is carrying that iron, it gives that characteristic purplish color, right? So when you run into the corner of your bed for the 16 millionth time, right, and you give yourself a big, nice bruise, what color is the bruise? It's blackish purple, right? That's because you have lots of heme that's been released because your red blood cells have burst, right? Yeah. All right. So heme is then degraded, and it's degraded in a sequential way over time. And so what happens to that bruise? It starts fading, right? And it goes from that purple to that gross, like brownish, greenish. It's like, yeah, right? Y'all know what I'm talking about. So that's the next intermediate. So as you look at your bruise and you can see it fades from that dark purple then to a lighter kind of greenish, nasty color. And then in the last stage, it's just what? Yellow. So yellow is bilirubin. So we talked about this really, really briefly before we did the case study. But where is bilirubin processed? In the liver. So if you have an inefficient processing of the bilirubin, then you're going to end up building up this yellow color throughout your entire body, right? So infants whose liver function is not all the way functional yet can be jaundiced, right? So what do they do for them? Put them under UV light, because UV light will break down bilirubin. So it kind of like gives their liver a break so that they don't have to use it. And then usually that's premature babies. But then once bilirubin is actually broken down, sorry, I'm like putting this all over the place. Eventually some of it goes to the intestines, some of it goes to the kidneys, different degradation products. So the degradation product that goes to the intestines is brown, which is why poop is brown. Then the degradation product goes to kidneys. Urobilin is yellow. That's why urine is yellow. So cool, right?
I didn't even know about that.
Yeah, yeah. You did. You did.
No.
All right. So we're going to talk for the rest of the period, well, almost the rest of the period, about tyrosine. Tyrosine is one of those super, super, super important biomolecules, right? So tyrosine is a precursor for lots of ketocholamines. So the really important ones, right? Dopamine, epinephrine, and norepinephrine. If you have a deficiency in dopamine, what disorder do you probably have? Parkinson's. Well, yeah, could be. But severe loss of dopamine is Parkinson's, right? So what's the characteristic of Parkinson's? Tremors, right? Lots of tremors. And so we're going to talk about how that happens and kind of some treatments too. But all three of these are important not only in neurosignaling, but also in metabolism, right? Because we've talked about epinephrine and we've talked about norepinephrine before, right? In Biochem 1. And what do we talk about with those? Right, all this is one of our fight or flight hormones that are released, right? Okay. So if we look at the normal tyrosine synthesis pathway, right? If you start with tyrosine, if it goes through tyrosine hydroxylase, what you're going to make is called L-DOPA. So L-DOPA is a precursor for dopamine. If you have somebody who has Parkinson's and they don't have enough dopamine, why can't you just give them dopamine? Yeah, but why? You're right. You give them L-DOPA. Why?
You get across the blood-brain barrier.
Dopamine can't cross the blood-brain barrier. You can give it to them, but none of it's going to get to the brain. So we give them L-DOPA. L-DOPA goes across the blood-brain barrier. Then the body can metabolize it into dopamine. Because a lot of times what happens is it's this enzyme that's no longer functional. So if you give them the precursor that skips this enzyme, then they can process it. It's not a perfect fix, and eventually too much L-DOPA will give you for a different reason will cause tremors. So it's not a perfect solution. So what they're trying to do right now is to actually implant stem cells into patients' brains that make dopamine. So that's kind of a big wave of the future, right? Okay, so once you have dopamine, then the dopamine is converted to norepinephrine, then norepinephrine to epinephrine, right? So all of this is all dependent upon tyrosine. So tyrosine is a really, 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 really important precursor. And so all of these signaling molecules bind to G protein coupled receptors to initiate all of their different pathways. No. Yeah, what receptor it binds to. It has a slightly different structure, so it binds to different things. They do basically the same thing. They do basically the same thing. Yeah, yeah. All right. So tyrosine is also really important in pigment formation. So when we talk about albinism, right? Albinism is the lack of pigmentation. So when you start with tyrosine, if you lose what's called Tyrosine converts one of the metabolic precursors that tyrosine turns into and prevents formation of the two different main pigment molecules. So you have eumelanin and you have pheomelanin. So eumelanin gives us that darker black-brown color, whereas pheomelanin gives you the red and yellow. So if you are a redheaded person, right, you have a predominant amount of pheomelanin and you have less eumelanin. So it's all about which one you have more or less of, right? If you have brown hair, brown eyes, you have lots of the eumelanin. It's all good. It's all good. Doesn't matter, right? All right. So if you have primary albinism, right? Primary albinism is an autosomal recessive trait. So you have to get 2 copies of the mutant tyrosine so that you don't make pigments. So now what are you more susceptible to? If you don't have eumelanin, you don't have pheomelanin, what are you more susceptible to? UV radiation, so cancer, right? So you have increased cancer. That's a big one. There are some other things you have to worry about, but the number one big one is skin cancer, right? So you have to be very careful about sunscreen, long sleeves, all that kind of stuff. What's that? Eaten. Yes, eaten very, very quickly. Absolutely. Absolutely. Exactly. Exactly. All right. The last little thing that we're going to talk about in this chapter is nitric oxide. Do you remember when we talked about nitric oxide in Biochem 1? A little bit, right? So if you have nitric oxide, let's do it up here kind of at the top just as a reminder, right? What is nitric oxide going to do? If we increase nitric oxide, do you remember what? Vasodilation. Vasodilation, right? So it's all about, remember, it was activated by the cyclic AMP. Do you remember those pathways? And we'll kind of give you the refresher in a minute. But nitric oxide is made from arginine, right? Because this is all about what amino acids can we use to make different things. So we need an enzyme called nitric oxide synthase, which is awesome. Can't we name them all this way, right? I know. So what we talked about last time was just the epithelial version of nitric oxide, which results in vasodilation. But there are actually 3 different kinds. So the endothelial does our vasodilation, right? There's a neuronal type of nitric oxide, and this just is involved in cell signaling in the brain. And then you also have an inducible version, which is in your immune cells. And we're not really going to talk about these last 2, but I want you to know they're there. But the thing with nitric oxide is it's very, very, very short-lived. It only lasts for a couple of seconds. And then it gets broken down very, very quickly. So it's a good response, but not a long-lasting response. So let's see what actually happens inside of the cell, right? Okay, so I'm gonna really zoom in. So we have some kind of neuronal input, right? And that releases acetylcholine from a nerve cell, right, onto our target cell. So this is an epithelial cell. We're talking about the endothelial version right now. So we're in a blood vessel, and this cell up at the top is the epithelial cell. And then we're going to communicate with a smooth muscle cell in just a minute. Okay, so we have our GPCR, right? Our GPCR becomes activated. What does it do? It exchanges GDP, GTP. Once we're activated, we activate PLC. What is PLC? Phospholipase, right? It cleaves lipids, phospholipase C. Phospholipase C works on PIP2, which converts PIP2 into 2-second messengers. What are they? Diacylglycerol and IP3. IP3, do you remember we said one of the effects of IP3 was that it goes to the endoplasmic reticulum and it mediates release of calcium, right? So IP3 goes, mediates release of calcium. That calcium will go and activate nitric oxide synthase. Nitric oxide synthase is regulated by a molecule called Calmodulin. We didn't really talk about Calmodulin. We touched on it super, super briefly. But Calmodulin is just a regulatory protein that responds to calcium concentrations, right? So the whole complex is basically inactive unless calcium comes in, binds to Calmodulin, Calmodulin activates nitric oxide synthase, right? Good so far? So once we have activated nitric oxide synthase, what's going to happen? We convert arginine to nitric oxide, and then the other byproduct is citrulline, but we don't really care about that, right? Nitric oxide, tell me about this molecule. Do we have to export it? Do we have to, what's up with it? It simply diffuses. It simply diffuses across the membrane, right? It's pretty much nonpolar, but pretty much diffuse across. All right. So if it diffuses across to a different cell, so if this is our epithelial, and this is our smooth muscle. So we're going to diffuse across to the smooth muscle cell. The nitric oxide binds to the guanylate cyclase, which converts GTP to cyclic GMP. Remember, just like cyclic AMP, cyclic GMP is a great second messenger, right? Once we have cyclic GMP will go and activate protein kinase G. right? Similar to protein kinase A, same difference, right? Protein kinase G will then work on its targets, and you don't have to know what its targets are, but eventually what's the downstream effect? Vasodilation, so muscle relaxation and therefore blood vessel dilation. What's the deal with cyclic GMP? How long does this float around? Long time? No. Very quickly, it goes back to GMP. So this is not a long-lasting effect. This is a very short-term effect. So we learned about it last time, right? That we wanted to, when we want to treat people who have heart disease, right? You come in and say, I think I'm having a heart attack. And they were trying to develop drugs to prevent that. They developed Viagra, right? And so what does Viagra do? It prevents cyclic GMP from going back to GMP. So you have maintained muscle relaxation and blood vessel dilation, right? So that didn't fix heart disease, it fixed a different problem. Now, when you go in to the ER and you're like, oh my God, I have this crushing chest pain, right? And you tell the doctor, I think I'm having a heart attack, right? And they say, okay, we're going to give you nitroglycerin, right? What is nitroglycerin? My grandmother used to carry her nitro pills. I need my nitro pills. Oh my God. But she really had lots of heart attacks. It was crazy. How long she survived after having so many heart attacks.
Nitric oxide attached to supplemented diffuse into the same muscle cells.
So nitroglycerin, very similar, glycerin, right? It's one molecule that actually breaks down into 3 molecules of nitric oxide. So nitroglycerin is also an explosive, right?
Yeah.
Because
that's why I was so.
you were like, what? Yes. Because you go from 1 nitroglycerin molecule to 3 molecules of nitric oxide, right? It's a release of a ton of energy.
And that's happening throughout that,
which causes the explosion. But in the drug that we take, right, the nitroglycerin is stabilized with cellulose, so that doesn't happen. So you're not your little old grandma who's carrying around her nitro pills. It's not going to explode. It's okay.
So it's stabilized
with cellulose. Cellulose.
It's only whenever it gets to the smooth muscle.
Okay, so let's go back. You have that patient who came into the ER, right? And they're crushing chest pain, classic signs of a heart attack, right? So as a physician, you want to give them nitroglycerin. What should you ask them? Are you taking Viagra? Why? Why can't they take nitro if they've taken Viagra? Okay, so Viagra will drop your blood pressure. And then you add on top of that nitroglycerin that drops your blood pressure even more. You are going to completely tank their blood pressure and you are going to kill the patient, right? So people lie when they come into the doctor's office. They do. So you have to explain this. If I give you both of these, did y'all ever see, what was the name of that movie? There was a movie where they did this. It was with Jack Nicholson in it. Oh, I don't remember now. And Keanu Reeves. I don't remember the name of the movie. You're right. So, but you have to be very careful that you're not doubling up on vasodilators and then causing some serious harm.
It's the same reason why you don't give insulin to a patient that's already hypoglycemic.
Correct. Exactly. Then you put them in a coma and they probably die. Right? All the things to avoid as a physician. Yes, please. All right, cool. Questions? Okay, that's the end of 17. End of 17. Holy moly. 18. 18 is really cool. Yes. That's awesome. All right, so in 18, we are going to talk about nucleotide metabolism. So we're going to talk about how we make our nucleotides. We're going to talk about how we break them down. We're going to talk about what we use them in, all kinds of stuff. And the little chapter opener in this one is really, really awesome because it talks about cancer treatments and explains it in terms of nucleotide metabolism. So what's a hallmark of cancer cells? Uncontrolled division, right? But the problem with cancer cells is not so much that they can grow uncontrollably, but they also grow uncontrollably and don't do any of their normal jobs, right? So it's like weeds in a garden. Eventually the weeds will take over the garden, right? If you don't do anything about them, it will. And that's why you'll die from cancer. So in order to continue to replicate, right? Every time the cell replicates, it also has to make its DNA. So you have a huge increase in DNA synthesis because you have an increased rate of cell division, right? So what we have done is we have made, what are the two main treatments for cancer? Chemo and radiation. What is chemo? Chemo is poison. It is poison. So is radiation. It absolutely is. But they are chemotherapeutics. So there are chemicals, right, that treat cancer. So here's one example of that, methotrexate. So this is an enzyme. This is a molecule that will inhibit an enzyme in nucleotide metabolism. So dihydrofolate reductase is an enzyme that is required in the synthesis of nucleotides. It actually is an enzyme that makes a cofactor, a coenzyme, not a cofactor, a coenzyme. And if you don't make this coenzyme, what do we know about enzyme function? No coenzyme, no enzyme function, right? Okay. So we make this molecule methotrexate. Methotrexate binds to this enzyme. It inhibits the activity of the enzyme and prevents nucleotide synthesis. If you stop nucleotide synthesis, what are you going to stop? You're going to stop basically all cells from dividing. How do you give chemotherapy? Do you inject it into a tumor? No. How do you give chemotherapy? Intravenously or a port, something like that. But you put it into the bloodstream, right? So it's going to go everywhere. So you are going to inhibit new cell formation basically everywhere. So what are your, yes, you're going to inhibit synthesis of cancer cells. Yes, absolutely. What else are you going to inhibit? And the biggest effect is going to be on the fastest growing cells. Hair. That's why people's hair fall out. What else? Nails. What more? Even more than that. Stomach lining. Because what are the symptoms of cancer treatment? Hair loss, nausea, and vomiting. Those are the big two. Those are the really, really big two. So hair follicles grow super, super fast. Think about your stomach lining. You make hydrochloric acid, right? And so the lining of your stomach cells secretes mucus to create that layer barrier between the hydrochloric acid and your stomach cells. But at the same time, you know, everything's kind of sloshing around. Those cells aren't going to live for very long, right? So they have a very, very rapid turnover. So if you stop making the mucus, protected mucus lining because your intestinal cells are not replicating, what are your symptoms? Nausea and vomiting, right? Yeah, basically. So I think it's really important to understand, you know, all of our cancer treatments are not specific to cancer. They are specific to a characteristic of cancer cells, but that characteristic is also shared by other cell types. Absolutely. So if you ask me what I think is going to be like the next wave of cancer treatments, it's going to be not only specific targeting to cancer cells, but I think it's also going to be where you take a sample of the patient's cancer cells out.
Personalize it.
And you absolutely personalize it, because that's the problem with cancer is not only is it like, if you have a bacteria, right, we can give you a drug that is specific to bacteria because bacteria are so different from us, right? No problem.
Cancer develops from ourselves.
But cancer is not only eukaryotic, but it's us. And so it has all of the external features of a cell that belongs to our body. And so the immune system has a really hard time differentiating them. And that's why a lot of them escape. So one portion of new cancer therapies is programming immune cells to better recognize cancer cells, right? Hijack our own innate, because we develop cancer all the time, but our immune cells typically kill it and are done with it, right? But sometimes it escapes and then you have a tumor formation and then you get cancer and all that, right? But so that's one side of it, enhance the immune system. And the other side of it is to take those cells out and then develop some way to conjugate a targeting mechanism to the chemotherapeutic. Then give the chemotherapeutic to the patient intravenously, but it's only going to the cancer cells. It's not going anyplace else.
Keep the normal cells healthy.
Yeah, so it would leave the normal cells alone. So the other thing that's a problem. Yeah. Oh yeah, absolutely. Absolutely. Try to stay on top of all this stuff. It's science is moving at a rapid pace. So sometimes chemotherapy stops working for cancer patients. Why?
Resistance.
Because it gains resistance. So if you have a cancer that accumulates a mutation, for example, leucine to arginine at residue 22 of this enzyme, will prevent this drug from binding to the enzyme. Now, the drug has much less of an effect. The cancer cells keep growing, but your normal cells did not develop a mutation to it. So they're still susceptible. So you're still going to get sick, but the chemotherapy is not working, right? So just because you have the symptoms of getting chemotherapy does not mean that, yeah. And so then you can have, if cancer cells become resistant to one drug, a lot of times they'll get multiple drug resistance as well. So it can get really, really difficult to treat certain cells, because then you want to keep upping the amount of drug. Yeah. Yeah, good. Okay. I thought that was a really interesting little intro, and we'll come back to it towards the end. So today we're going to talk about structure and function of nucleotides, and really why it's so important to recycle them. Then we'll do purine and pyrimidine metabolism, and then we'll talk specifically about DNA, deoxyribonucleotide metabolism. All right, so we already have talked about nucleotides being present in, oh my gosh, so many things, right? Energy conversion reactions. Tell me about energy conversion reactions. Where have we used nucleotides in energy conversion reactions? Which one comes to mind? Which nucleotide? ATP, right? That's our energy currency of the cell. How many different reactions have we used ATP in order to drive a reaction?
All of them almost.
Like almost every day. Okay.
And
GTP also, but the main one is ATP, but GTP also, you're right.
It's basically.
Yeah, it's basically ATP. All right, signal transduction pathways. We talked about one like 5 seconds ago. Cyclic AMP, cyclic, well, which one? We didn't talk about cyclic GMP, right? So on and so on. Okay. Coenzyme dependent reactions. FAD, NAD, right? NAD plus. Okay. Genetic information storage and transfer,
Like all
DNA and RNA. Okay, good. That was the easy one. So are they important? Yeah, pretty much. Okay. So try to remember back in Biochem 1, our nucleotide structure. If we have a nucleotide, what do we have? We have a base, a sugar ribose, and phosphoryl groups, right? Depending on how many phosphoryl groups, you'll be ATP, ADP, AMP. All right. Talk about our sugar. What's the difference in sugar between RNA and DNA? Nice quick reminder.
OH versus no OH for
So if it's ribose on C2 prime, why do we do this prime business? It's so you don't get confused with the atoms that are in the base. So whenever you see primes are always referring to the sugar. If you see C1, then you're referring to the base. Okay. So RNA has hydroxyl, deoxy has no oxygen, just a hydrogen. Right? Okay, so purines and pyrimidines. Purines are pure as gold. I guess I should capitalize. Pure as gold. So what are they? Adenine and guanine. Right? So you remember them, pure as gold. Then our pyrimidines, cytosine, thymine, and uracil. Okay. Which ones are in DNA? Adenine, guanine, cytosine, and thymine. Uracil is in RNA only. Is uracil in RNA because it is less stable? The answer to that is no. It is all about proofreading. We kind of talked about this in Biochem 1, but I feel like this is a really great place to reaffirm and to make sure that that misconception is gone for y'all. So the whole reason that we have uracil in RNA only is a proofreading mechanism. So if we have cytosine, if cytosine is deaminated, what's it going to turn into? Uracil. So if cytosine is deaminated, it goes to uracil, right? Okay. If I start out with a base that is, what's my complementary pair to C? CG, right? Okay, I have a CG pair in DNA, right? That's my normal pair. My cytosine gets deaminated and it goes to uracil. Uracil now pairs with A. I go through one round of DNA replication. What's my new pair? So what did I just do? I did a site mutation. I went from a CG pair to an AT pair. That's a problem, right? Okay.
Which is why we have MMR.
Why we have what?
MMR.
So how do I prevent this from happening? Is cytosine going to spontaneously deaminate to uracil? It happens. It will happen. So what does the cell do in response? I have enzymes. We all have enzymes, not just me. We all have enzymes. Me specifically, yes. I have enzymes that are going to go in and remove any uracil that is present in the DNA, right? It's a proofreading thing. Every time the DNA is replicated, I have all these extra proteins that are going to go in and make sure that everything is right. And if it sees a uracil in DNA, what's going to happen? Get rid of it. It's going to get rid of it and replace it with a what? With a C.
Because that's the only thing that deaminates to you is C.
The only thing that deaminates to make you is C. So anytime it sees a U, it replaces it with a C. Otherwise, we'd have an insane amount of mutations and it would be crazy. We're going to talk about there are two other nucleotides that you're going to need to know. One's a purine, one's a pyrimidine, and they are precursor molecules, but we're going to talk about those in a bit, but you can go back and add them when we get to them. All right, so the book did this really nice little job in putting together all the different functions of nucleotides, right? So, and we kind of talked about this in the beginning, excuse me. So ATP just as a representative nucleotide. ATP is used in energy conversion. What's the example they're using here? There's ATP in a glucose molecule. What's happening? What is this enzyme? Hexakinase is phosphorylating glucose to glucose 6-phosphate and trapping it in the cell, right? Okay. Signal transduction. So in signal transduction, we have a G protein coupled receptor, right? And they're doing this with a beta-2 androgenic receptor through glucagon signaling. But we activate our G protein coupled receptor, we activate adenylate cyclase. Adenylate cyclase converts ATP to cyclic AMP, and we initiate that phosphorylation cascade. In this case, it's the pKa. But that's a common thing that y'all already know, right? Okay. Coenzyme function. If we're going to talk about ATP, which coenzyme are we going to talk about? Something with adenine in it. So NAD has adenine, dinucleotide in it, right? And then genetic information we already know, right? All good. Okay. This nucleotide salvage pathway is really, really important. If I had to go in, if our bodies had to go in and make nucleotides from scratch, it would be extremely, extremely energy inefficient. And so what you're going to see is that the body is a great recycler. And so it has this amazing salvage pathway. So we're going to do this model using RNA, because if you think about the two molecules, which one, RNA or DNA, is going to hang out in the cell longer? DNA is going to hang out longer. RNA has got to be turned over very rapidly, right? Because it's going to be, we're going to go from DNA to RNA, ship our RNA over to the ribosomes. Ribosome's going to go make protein, and then that RNA needs to be degraded so we can go again. So just think how many nucleotides you would have to make from scratch. A lot. So we have to have a really great way to salvage it. So this nucleotide salvage pathway drawing is based on RNA, but it also works in almost exactly the same way for DNA. So don't worry about that. All right. So we're going to start out, and we have an RNA strand. So one thing you have to remember, how many phosphates are attached to each nucleotide in a built RNA or DNA molecule? Per sugar and base, how many phosphates do I have? Only one. So in the RNA strand, I have a nucleotide monophosphate. A lot of people get confused and they're like, why do we only have one? Well, because in the DNA or RNA, you only have one. The next phosphate up here belongs to the next nucleotide, right? Okay, so we go through a first round of RNA degradation. So we have endonucleases that go in and chop up the RNA. Now, we're going to get oligonucleotides. What are oligonucleotides? Short, less than 10 nucleotides per strand, right? Now, once we have oligonucleotides, we're going to undergo a second reaction that is phosphodiesterases. Phosphodiesterases use water to break apart individual nucleotides. So all of these nucleotides, they're going to be, nucleotide triphosphates, diphosphates, or monophosphates. Nucleotide monophosphates. So like AMP, right? So you know why I put the N instead of like an A? Because N stands for any nucleotide. So we have NMPs. Once we have individual nucleotides, those individual nucleotides can be phosphorylated, right, through kinases, and we're going to make nucleoside triphosphates. So now we have 3 phosphate groups attached. This is the substrate for RNA polymerase. DNA polymerase too. Once we have 3 phosphates attached, now we can go on and add them to the new RNA that we are making for whatever protein we want, right? So this is extremely energy efficient. We break them down to the individual nucleotides, attach some more phosphates, go again. So the energy investment is very low, very low. You're going to use those endonucleases to make oligos. Then you use the phosphodiesterases to make individual nucleotides. You phosphorylate the individual nucleotides and put them back into an RNA. Good so far? Okay. Now, this bubble fits right under the previous bubble. Do you see what this is? A nucleotide, excuse me. So this is a nucleotide monophosphate. What was at the bottom of the previous circle? A nucleotide monophosphate. So the second circle fits right here. Y'all understand what I'm saying? Okay. So sometimes you want to salvage just the base. So what you do is you have your nucleotide monophosphate. If I want to keep the base, what I need to do is first cleave off these phosphates, right? So I'm going to convert from being a nucleotide to being a nucleoside. So a nucleoside, the only difference is here we have a phosphate, here we have a hydroxyl, right? Okay, so the next thing is that I want to cleave off the base. How am I going to cleave off the base? I basically just replace the base with a phosphate group. So now I have a sugar that is phosphorylated, but let me ask you this. Is it phosphorylated on the same carbon where it's phosphorylated in a nucleotide? No, it is not. We're going to talk about why that's important. Okay, so if I replace the base with an inorganic phosphate, I'm going to release the base and I'm going to release a phosphorylated sugar. Let's first track the base and then we'll talk about the sugar. So once we have just the base, I can attach this to what's called PRPP. PRPP is phosphoribosylpyrophosphate. So if it's a pyrophosphate to have two phosphates hooked together. So the two phosphates that are hooked together are on the same carbon that the base will eventually be on. Why is this really important? This is an energetically smart way to run this reaction. Because if we replace the base with the pyrophosphate group, the pyrophosphate group is kicked off. Now tell me what happens to pyrophosphate. Pyrophosphate splits into two inorganic phosphates. This reaction right here is energetically what? Energetically favorable. So it has a very low delta G, very negative. We'll put not low, we'll put negative. So we can drive that reaction. So you're going to hear PRPP often. So the other, why do we call it PRPP? Because follow the molecule. Phosphate, ribose, pyrophosphate. So it's phospho, ribosyl, pyrophosphate. It's a really great name. I feel like sometimes they make this really great. It's very good. It is. Okay. So let's go back to our sugar over here. Our sugar over here, this is ribose 1 phosphate. Can I just use ribose 1 phosphate to attach a new base? No. No, because the growing chain of phosphate needs to be over here on carbon 5, right? So this is carbon 1, this is carbon 5. So we need to have a phosphate here so we can make ATP, AMP, ADP, right? So we're going to start out with ribose 1 phosphate. But let me ask you another question. Where can we get ribose 1 phosphate? As animals, where can we get ribose 1 phosphate? We're going to have to pull from the dredges. Pathway. Pentose phosphate pathway. We could pull ribose 1 phosphate from the pentose phosphate pathway. What if we're plants? You can build it, right? This is one of the sugars that you could build from. Yeah. But we're not plants, so it's okay. I don't know. I think that would be a very unsatisfying way to eat. Sunlight, I just can't. Okay, so then we have ribose 1 phosphate, but what we need is ribose 5 phosphate. So we go through a mutase enzyme, and all that happens, right, is that we swap the placement of the phosphate and the hydroxyl. Now, this is a substrate for PRPP synthase. PRPP synthase is going to use ATP in order to phosphorylate what? The hydroxyl, well, the carbon what? Carbon 1. How are we doing? Pretty good? It's a lot. Okay. And that's, so what you might kind of want to do is to sit down and draw these pathways hooked together, right? So you can understand and see the difference between base salvage and then nucleotide salvage, right? So you can see the difference. And you can see what feeds into what. You do need to know, you don't need to know necessarily exact enzyme names other than PRPP synthase. This one you need, because we're going to talk about PRPP synthase again. But you need to know the type of enzymes, right? So if you come up to the top, you need to know this is an endonuclease, right? Here's a phosphodiesterase. Here's a kinase, right? We're not naming these individual enzymes because it's more important that you understand what class of enzyme is required, because the enzymes will differ if it's RNA synthesis versus DNA synthesis, right? So you just need to know the types. Okay. And that's where I'm stopping today. I'm calling it today. I think we've had enough. I think we're full. Yeah. Full of knowledge. So this chapter is super, super, super interesting. It's as close to as we get to like DNA and all the really, really interesting stuff that goes along with that. I don't know if I can pick my favorite, if I love proteins more than DNA and RNA, but I don't know. It's all awesome. It's all awesome. Did y'all hear? This is real. Did y'all hear they finished completely sequencing the human genome?
I was actually to read about that.
Yeah, so you should really read it. So here's the deal. Why we've been around for so long. We started this project, genome sequencing. This started when I was in like elementary school. And so I picked it up when I was in high school, and I picked up books about it, and I was like the biggest nerd ever about it, and I read all about it, and I wrote papers about it, and you know, my English classes, and my English teacher was like, I don't understand half the words that you put on this page. Just loved it. It's what got me into biochemistry, really. But the problem is that the techniques, it's all a technique thing. The techniques that we used were unable to read accurately the repetitive sites in DNA, right? So there are lots of portions of the DNA that when I was growing up was called junk DNA, right? Did y'all hear that? Y'all, anybody say that in front of y'all, junk DNA? Yeah. Bull crap. None of it is junk. And there was one thing that always got me when I would read all this. I'm like, how can so much of the DNA be junk? It can't just be junk. It just can't. And so the number of repetitions, what composes the repetitions, will actually control how DNA is processed, how DNA is folded, how DNA is transcribed. I mean, it's unbelievable how many control mechanisms are present in repetitive DNA. And so the techniques were unable to read a long enough sequence to see exactly how long and what the composition was of these repetitive elements. So that's the 10% portion of the genome that we could never perfectly sequence. And so with new technology, I think now they said they can go up to 100,000 bases in a row. And so with that, they can successfully sequence really long repetitive parts of DNA, which I thought was insanely awesome and cool and wanted to share. Okay, that's my rant. That's my rant. Go ahead. I'm sorry, I said I would let you go and then I held you. Yeah, you're done. You're done. Oh, you're done. Go. If I just want to keep chatting.
That's kind of big.
Yeah, it's amazing. It's absolutely, no, like this was what got me. This is what hooked me in science. I feel like, you know, things like this, that when it brings science full circle, it's totally amazing and wonderful.
And now finally we get to like process and always see the entire, someone's gonna make a poster.
Well, now, okay, you've sequenced it, but is it different in different cell types in terms of number of repetitions and what part of the DNA is euchromatin versus heterochromatin? Like there are 10,000 other questions that this opens the door to.
It's gonna take more years to see.
Oh yes, it's never ending. Science is, that's the beauty of science. It is absolutely